So let's wrap this up, part 4. So human traits. We've talked a lot about the examples of the pea plants that Mendel worked with. And so what about humans? So humans are not good subjects for genetic research. It's kind of contrary to why the plants were such a good, model for being a subject for genetics. Humans, they don't have those advantages. The generation time is too long. Parents produce relatively few offspring, and breeding experiments are unacceptable. Like, it's unethical, basically. But we can study genetics from the perspective of existing family and generations. So, basic Mendelian genetics is still the foundation of human genetics, and we can still kind of study it just with the information that we already have about family members and different generations. So in human genetics, geneticists analyze the results of human matings that have already occurred. So geneticists will find out more information about a person's family in different generations and be able to map out, certain traits and see how they got passed on. So a pedigree is a family tree that helps describe the inheritance of a trait across generations. So usually you're focusing on one trait at a time, and you can see how this trait gets passed on in a family, and again, one that's like already existing. So this is one example. Here are another couple of examples. So this is the same family tree, but you're focusing on two different traits. So, or characters, as Mendel said, but so you're focusing on two different things. So it's the same family tree, but the shaded in shapes show that particular trait that you're focusing on. Squares represent males and circles represent females. And this is true of all pedigrees. They're standardized that way so that you can always interpret them. So on the left-hand side in panel A is focusing on a widow's peak. So a widow's peak meaning like if a person's hairline comes to a point in the middle, that's a widow's peak. And if there isn't one. So those are the two alleles for this thing. So everybody that has a filled in shape on this panel is, has a widow's peak. So they can track, okay, this person did, this person did, this person did, this person did. And so this is a couple. And so they're tracking each half of the family that is part of this mating. And then so these are the two daughters of this couple, this mate. So when they're connected to each other, that is like a mating. And then branches that come down are, offspring. So this couple had these offspring, and then if we track each half of that family to see how that gets passed on, so we see some family members have the widow's peak and some don't. And so in this example, a widow's peak is a dominant trait. And then on the right-hand side is a totally different trait. Again, same families, but you're focusing on a totally different trait. And in this case, it's whether or not you can taste this chemical that's called PTC. It's a little bit of like a bitter taste. This is also a lab that can be done in person. It's really interesting. And so if you taste it, then that is also dominant. If you cannot taste it, that is recessive. So the people that cannot taste it, we're actually focusing on that, like who can't taste it. So the squared, the shapes that are filled in are whether or not you can't taste this. And so it turns out that this is recessive, not tasting PTC. And if you can taste it, then that is dominant. So if you can taste it, you don't know if you're homozygous dominant or heterozygous. And same for this widow's peak. You know that you have at least one copy because there's a widow's peak, but you don't know if it's that you have both copies or one dominant and one recessive copy, and that's just always going to cover up that recessive. And so pedigrees can be used to make predictions about future offspring. Again, each offspring is just going to have a particular percent probability of maybe having this trait. It's not like every 4 kids is always going to have this 3-to-1 pattern. Again, every single offspring is going to be its own chance, its own independent event. So it's just kind of helping you map out and estimate, maybe make predictions about future offspring. So again, we can use the multiplication and addition rules to predict the probability of specific phenotypes. And there are different disorders that are passed on that are genetic, and there are some that can be recessive, and there are some that can be dominant. So many genetic disorders are inherited in a recessive manner, and these range from relatively mild to life-threatening. And so recessively inherited disorders show up only in individuals that are homozygous for the allele. So remember, homozygous, recessive. So parents can be carriers. They can carry that recessive allele but not know because it's not going to be expressed. So carriers are heterozygous individuals who carry the recessive allele. They have a dominant and a recessive version. But because this disease or disorder is recessive, it'll only be expressed if you have both copies recessive. So carriers are phenotypically normal, but they are carrying that recessive allele. So most individuals that have recessive disorders or conditions are born to carrier parents. The parents had to be carrying the recessive version and each give a recessive copy to the offspring. So for example, albinism and cystic fibrosis are both recessively inherited. So here's an example. 2 parents can be carriers of the trait of albinism, but, be phenotypically normal, but they're carrying it. And then so if they both happen to be carriers, there is a 1 out of 4 probability that an offspring can have albinism. If there is a recessive allele that causes a disease and it's rare, it's unlikely that 2 carriers will meet and mate. It's kind of like up to chance, but it's pretty unlikely, small chances that 2 carriers will meet and mate. Consanguineous matings between close relatives increase the chance that both parents will be carriers for that same rare allele. But most societies and cultures have laws or taboos against that. So we don't see that often anymore. It used to be a lot more common in like royal families, they would try to keep marriages within the royal family. So things like, oh my gosh, the bleeding disorder, hemophilia, that was common in one of the royal families because it's a recessive inherited disorder. And because people within that family would mate, they would be more likely to get that rare allele. But yeah, so we don't see that as much anymore. It's kind of just a smaller percentage that it would happen. So most human disorders are recessive. Some are caused by dominant alleles. Dominant alleles that cause a lethal disease are usually rare and arise by mutation. And some examples of dominant inherited traits or disorders are achondroplasia and Huntington's disease. So achondroplasia is also known as dwarfism. And so it's actually a dominant trait. Again, a trait being dominant or an allele being dominant doesn't always mean it's more common in a population. It kind of just depends on the trait. So it is a dominant trait. As well as Huntington's disease. And then Huntington's disease is different because it's like a phenotype that hides for a while. So the timing of onset of that disease or any other disease like really affects how it's inherited. If somebody is going to pass away before they can even reach you know, the age to be to pass on their genes, then it's not going to get passed on as much, you know. But if something like Huntington's disease comes later in life, then you won't know until it's a little bit later. So Huntington's disease is a degenerative disease of the nervous system. And so the disease has no obvious phenotype until the individual is about 35 to 40 years of age. And so then some degeneration of the nervous system starts and then continues. Once the deterioration of the nervous system begins, the condition is irreversible and fatal. There is a test that can detect the presence of the Huntington's allele in your genome. Some individuals with a family history of Huntington's disease choose to be tested for the allele, and others decide that it would be too stressful to find out, so they kind of just live and see whether or not they develop it or not. And so those were just some examples. There are many diseases such as heart disease, cancer, alcoholism, and mental illnesses that have both genetic and environmental components. So again, it's not just genetics, you know, nature and nurture also play a part. The nurture, like your environment, can play a part in all of these diseases, you know. Heart disease, cancer, alcoholism, mental illnesses, like it does matter your lifestyle that affects development of some of these, not just genetic predisposition. So no matter what our genotype, our lifestyle does have a tremendous effect on phenotype. And genetic counselors can provide information to prospective parents concerned about a family history for a specific disease. So yeah, if a family is concerned about Huntington's disease, they can meet with genetic counselors to examine their pedigree more. And fetal and newborn testing can also reveal genetic disorders. So for a growing number of diseases, tests are available that even identify carriers and help to find the odds of having an affected child more accurately. So even before, you know, fetal testing to see if you are a carrier. And so this helps enable people to make more informed decisions about having children. But they also raise other issues, like whether affected individuals fully understand their genetic test results and how the test results are used. For example, like, will that affect health insurance? And some examples of fetal testing. In amniocentesis, the liquid that bathes the fetus is removed, a little bit, not all of it, and tested for certain genetic disorders. There's another method called chorionic villus sampling, and that samples the placenta. And then other techniques like ultrasound allow the fetal health to be assessed, usually in utero. So you're not like extracting something, you can kind of just take a look and see what you can see. Here are some examples of those: amniocentesis, withdrawing fluid surrounding the fetus, or chorionic villus sampling is sampling through the cervix. And so that is still during development and gestation, pregnancy. But once a baby is born, they can also be tested. So some genetic disorders can be detected at birth by simple tests that are now routinely performed in most hospitals in the U.S. One common test is for this disease disorder called PKU, or phenylketonuria. It's a recessively inherited disorder that occurs in every 10,000 to 15,000 births. So that's just one example. They kind of like prick the bottom of the foot and test. But there's a lot more that they can test for. That's just kind of like one big example. The number of conditions that can be tested in newborns is over 100 and increasing. Okay, so that is everything. 4 parts. So hopefully this is what you can do by the end of studying this lecture, be able to explain Mendel's two laws of inheritance, use examples to show how genetic inheritance patterns can be affected by complete dominance for sure. And then also maybe a little bit of the other stuff too, like incomplete dominance, codominance, multiple alleles, pleiotropy, epistasis, and polygenic inheritance. And be able to explain what a pedigree is and use examples to show how human traits can follow Mendelian patterns of inheritance.